Next, I want to talk about how glycogen metabolism is controlled, because let's think about this for a second. If we have glucose 6-phosphate that can be made into glycogen, as we've been talking about, or that glycogen can be broken down into glucose 6-phosphate, right on one side we've got the glycogen synthase here, and on the other side we would have the glycogen phosphorylase. You can imagine if both the phosphorylase and the synthase were highly active, you would just kind of be going around in this circle. And remember, every time we add a molecule of glucose to glycogen, we consume a UTP molecule. So we would just be spinning around here uncontrolled, and that wouldn't be very effective. So what needs to happen is, realistically, only one of these enzymes needs to be active at a time, and the other one would be turned down. So only one active at a time. And so there are things that we would call allosteric controls. And so remember, we have looked at this with phosphofructokinase, or PFK, and the way that ATP levels influence that particular enzyme in glycolysis flux. Well, guess what? Here in glycogen metabolism, ATP and AMP, ADP, all of those also play a role as well. So here's one true thing as far as the synthase is concerned, that it is activated by high concentrations of ATP and or glucose 6-phosphate. Now, does this make sense? Kind of think through these types of things logically. ATP is, the energy currency of the cell, and glucose 6-phosphate is that intermediate in glycolysis. And so if both of these are high, or either one of those are high in concentration, that's kind of a signal that the cell has plenty of energy, that it has plenty of fuel, it makes plenty of ATP. And so as a result, it doesn't need to make any more. And in fact, it probably makes sense to store it away. So high levels of the energetic compounds, if you want to think of it that way, will activate glycogen synthase to store away energy in the form of glycogen. And so conversely, glycogen phosphorylase is activated by low concentrations of ATP and glucose 6-phosphate, because if ATP levels are low or glucose 6-phosphate levels are low, that means that the cell needs energy. It's low on ATP. We need to run glycolysis and other things to make more energy. And if there's not a lot of glucose 6-phosphate, that must mean that it's not coming into the cell from the bloodstream, for example. And so it makes sense that the cell should break down the glycogen that it has to make more glucose 6-phosphate. And that would make sense that the glycogen phosphorylase enzyme would be activated.